See, it's sort of the same sort of thing. Some proteins getting misfolded and tangled up inside the nerve cell, but it's different proteins. It means they have different modes of chemical action on them. But sometimes, to differentially diagnose without knowing what the actual protein is, Lewy body dementia often affects the substantia nigra, which is what is damaged in Parkinson's disease. So often with Lewy body's dementia you also get Parkinsonian symptoms, and that often gives it away. It's up to the clinicians to work it out. So a clinician has to make a diagnosis on the basis of only the behavioural tests and cognitive tests done. Can't take a blood sample, I'm afraid. And the only way to be ever sure is when the patient dies, they can have an autopsy on their brain, chop it up, and then actually do analysis on the neurons themselves and see what protein caused it. But this is useless because the person's dead. There's no hope of treating them now. The only significance of doing that is for research purposes, to find out what protein caused it, and perhaps find some medication that can sort of untangle the protein or get rid of it or destroy it. It's being done as we speak now. See, that's a major limiting factor in the treatment and diagnosis of many psychiatric and neurological illnesses. Because the ones you talk about, cancer, AIDS, diabetes, etc., they're all disorders of the blood, or something to do with the blood. So you can take a blood sample and find out the abnormalities in the blood. The ones we're talking about here are abnormalities of the brain. You can't just take brain tissue out and test that. If you could, then there's your biological markers. Only time you can do that is when the patient's dead. Or hopefully some metabolites from the brain come out and enter the bloodstream, highly unlikely. The blood-brain barrier prevents many things from getting into the brain and also getting out of the brain that we would want to test, such as these chemical imbalances that people so talk of. Another good way to do it, I guess this is the best way, is to take a brain scan and have a look at the structural differences. Then again, structural differences don't tell us everything. Functional imaging is a favourite. What you basically do is you put a patient or whoever into a brain scanner, and then what the machine does is measures the amount of glucose that's being metabolized in the brain. And it's sort of the hypothesis is that the area that's using the most glucose, that's using up the most glucose, is the brain area being used at the moment or in the task. And if it's sort of stays that way generally, because the whole brain's using glucose, of course, it's the amount difference which is looked at. If it's the same over time, and you can look at the abnormality of these things. Looking at sort of brain imaging studies and depression, increased glucose metabolism or increased blood flow to the amygdala is quite common, my favourite part of the brain. And also in the anterior cingulate, and also in various parts of the frontal lobe of the brain, increased blood flow to the medial orbital gyrus. Problem is, we don't know why there's abnormal activation of those areas. Well, it makes sense that amygdala is the fear centre and such, but you know exactly why, the detailed mechanisms right down to the very atom. But then again, diabetes type 2, it is unknown why the receptors are downregulated and insulin can no longer use those receptors anymore. Why? I mean, sure, the increase of sugar in the diet means there's an increase of insulin, which means the cell will downregulate the receptors to stop over-sensitization. But why is it that some people get diabetes type 2 and others don't, even though they eat the same amount of sugar? Same sort of thing in medicine. There's a lot of questions to be answered. A lot of studies also found that the hippocampus in depressed patients, the volume is smaller, so there's been significant atrophy happening there. Problem is with functional imaging studies is that a lot of studies find different results. But there's definitely some consistencies in there, such as the amygdala and the medial orbital area and the anterior cingulate. Problem is also that depression itself is not an illness but a symptom, and because it's a symptom it's caused by different illnesses. So, well, it's more known as depressed mood, I guess, and it also has its own disorder in the DSM called major depressive disorder. There's also minor depressive disorder, not in the DSM. And it's also a symptom of bipolar disorder under the mood disorders, or there's mood disorders not otherwise specified. Depression is also a symptom of such illnesses such as Cushing's syndrome, which is an abnormal production of cortisol. There is indeed a biological marker for depression that can be found in the blood. It's abnormal levels of a certain hormone, and this hormone's name is cortisol. But why isn't this diagnostic of depression? Because abnormal release of cortisol is also a symptom of many other illnesses. So you need to use this marker as well as everything else to have a good diagnosis of a depressive disorder. And then you have to diagnose which depressive disorder. So I guess I'll talk about how cortisol works and why it goes wrong in depression. This up here is a schematic of the hypothalamic-pituitary-adrenal axis. And this is what's a little bit broken during depression. Okay, so what we have here is abnormal activation of the limbic parts of the brain, such as the amygdala. Amygdala has strong connection to the hypothalamus, activates this, and it will release a releasing hormone, CRH, corticotrophin releasing hormone, which will activate the anterior pituitary gland and cause it to release ACTH. This ACTH travels in the bloodstream, goes to the adrenal gland. The adrenal gland releases a stress hormone called cortisol. It's a stress response, and you normally get it within an hour of a stressful event happening, normally, and then it's suppressed. And how that happens is it works as negative feedback. So the release of cortisol itself tells the hypothalamus to stop releasing CRH. It also tells the anterior pituitary to stop releasing ACTH. So, therefore, no more cortisol is released, so it just doesn't keep going and going and going. Unfortunately, in depressive illnesses, this stress state is perceived, and the hypothalamus keeps getting stimulated, and also there is a problem with the cortisol negative feedback. It doesn't work.